All right, well, welcome to the first lecture for PEPROF 202, Structure and Function 2. So I'm thinking we're going to end up dividing the cell chapter, which is chapter 3, into 3 parts. This first part is going to run through tonicity, and so we'll talk about osmolarity and cell size and water movement and all that kind of stuff in this first one. And then in the second lecture, we're going to go through the organelles. And then in the third lecture, we're going to cover DNA transcription and translation. So speaking of cells, without further ado, cell theory, as you can see there, is that the cell is the structural and functional unit of life. And so we're built of trillions of cells, but among those trillions of cells, we have a variety of different cell types. And so, for example, in the lower right, in this little figure, you can see a bunch of different cell types, whether we've got fibroblasts that help make up our connective tissue, or erythrocytes, better known as red blood cells that carry oxygen. Epithelial cells are cells that are effectively lining cells, so your skin is comprised of epithelial cells, but those epithelial cells are also found lining your gastrointestinal tract, so like inside of your cheek, down your esophagus, into your stomach, etc. And then, you got skeletal muscle cells, nerve cells, etc. And so among those 250 different types of cells, many of them are very small. So for example, our erythrocytes, our red blood cells, are only about 2/10,000ths of an inch in diameter, so they're pretty tiny. As opposed to some cells can be fairly large, or at least fairly long. So with our little nerve cell down here, so the term for a nerve cell is a neuron. And so, for example, if you've got what's called an alpha motor neuron, which is a nerve that innervates skeletal muscle cells, those neurons can be fairly long because the cell body of this neuron lives in your spinal cord, and then it has this axon that runs all the way out to whatever muscle fibers we need to innervate. And so if we're talking about a motor neuron that innervates muscles in the foot, well, then the cell body is going to be in your spinal cord, in the lower part of your spinal cord, so kind of down in your low back and a little bit below that. And then this little cord, there we go. This little, it's called the axon, but that's going to leave your spinal cord and run all the way down to those muscles. And so the length of that cell, of that alpha motor neuron that innervates the muscles of the foot, may be 3 feet long if you're of fairly normal stature, or if you're somebody like Giannis Antetokounmpo and you're exceptionally tall, that axon may be well over 3 feet long, right? So you can get fairly large or fairly long cells. So one of the things to understand there is the relationship between structure and function. So structure and function, obviously the title of this course, the structure describes basically how something looks, right? So it's how an organ is made or how it's comprised, and then the function is what does it do. And those terms are analogous to anatomy and physiology. Anatomy is the structure of an organ. So right, if we're talking about the anatomy or the structure of the heart, we're thinking about the fact that it has 4 chambers, you've got 2 atria, 2 ventricles, etc. So that's kind of the basic anatomy or the basic structure of the heart. And then the function is what does it do? Well, based on its anatomy, so you've got those 2 atria up top, 2 ventricles down below, and then you've got one-way valves between them. Based on its anatomy, blood can only flow one way. And if it doesn't, let's say you've got a problem with one of the valves in the heart, that's an anatomical or structural problem, and then that alters the function of that organ. So with the cells, their structure is related to their function. So their shape is related to what it is that they do. So for example, with our erythrocytes, with our red blood cells, you'll notice there that they kind of look like, if you've ever gone tubing down the river, they kind of look like that. They're not hollow in the middle, but they are fairly narrow right in the middle of the cell. So they're kind of these thin little discs. And so in terms of what they do, again, their primary job is to carry oxygen. And the reason that they're fairly flat little discs is we want to minimize the distance that oxygen has to move. As opposed to if they were big balls or big cuboidal shaped cells, kind of like this, then that would be farther that oxygen had to diffuse, and so they would just be less good at being able to pick up and offload oxygen. They would be less good at their job. And so their shape, the fact that they're flat little discs, enables them to be really good at both onboarding or onloading oxygen and then offloading it when it gets to the tissues where they need to drop off their oxygen. Same kind of thing here with our epithelial cells. So as mentioned, epithelial cells are lining cells. So for example, you've got epithelial cells in your small intestine. That's where we do most of the absorption of the things that we eat. So if you, some of the sugar that you take in will actually move across from one side of the epithelial cell to the other. And so they're built to move things from one side of the membrane to the other, and they're fairly thin. If they were much thicker than that, well, that's again a farther distance that glucose would have to move, and so we'd just be less good at glucose absorption. So there's a lot of interrelationship between the structure of a cell or the structure of an organ and what it is that it does, so the function then. So even though you've got all these different kinds of cells that are shaped in a variety of different ways, there are some general characteristics that they share. And so we're going to talk about kind of this concept of a generalized cell. So your generic or generalized cell has three characteristics. You've got a plasma membrane, which is sort of the outer border of a cell. So it separates the cell from its environment. That's your plasma membrane. And then you've got the cytoplasm, which is the kind of jelly-like material that's primarily water that fills the inside of the cell. And then you've got the nucleus of the cell. And the nucleus of the cell contains the genetic information. So we find our DNA inside of the nucleus of the cell. And one of the ways that I want you to think about DNA is as the instructions for building proteins. And I'll return to that theme and I'll mention that over and over and over again. But I think that's a really helpful and important way to think about what DNA does. So again, the instructions for building proteins. All right, so a couple terms on there. We're going to talk, at least in this first lecture, quite a bit about intra versus extra. So anything intra, I know I'm starting with the second term, anything intra is inside of. So for example, here in a little bit, we're going to talk a lot about intracellular fluid. So intracellular fluid is fluid inside of the cell. And of course, that's as opposed to extracellular fluid. Anything extra is outside of. So extracellular fluid is fluid that's outside of the cell. So anything intra is inside of, because we're going to use it in terms of intracellular. So anything intracellular is inside of the cell. Anything extracellular is outside of the cell. So in terms of extracellular material, so things that are outside of the cell, we've got extracellular fluids. So again, fluids outside of the cell. One kind that you can see there is the interstitial fluid. So inter as opposed to intra. So again, intra is inside of. Inter is between. So interstitial fluid is fluid between cells. So the way that this textbook describes the interstitial fluid is as a, quote, nutritious soup, end quote, that our cells are submerged in. And so in that nutritious soup, we've got things like amino acids, which are the building blocks for proteins. We've also got sugars, we've got fatty acids, and then we've got their waste products. So those waste products might include things like carbon dioxide and a host of others that the cell needs to get rid of in order to be able to continue its normal duties. Similarly, in terms of the amino acids, so the cell will have to take up things from its environment, so like your amino acids, again, the building blocks for proteins, so that it can repair itself, for example, is one of the things that it'll do with those amino acids. We've also got blood plasma. So blood plasma is the fluid that your red blood cells, your erythrocytes, float in. And so blood plasma goes up or down depending on your hydration status. So if you are really well hydrated, then you end up with an increased blood plasma volume, and effectively your blood is thinner. It's yeah, we'll leave it at that for now. So your blood's a little bit thinner. And then if you get dehydrated, your plasma volume goes down, so the blood effectively gets thicker, and then it gets harder for the heart to pump it. And we'll talk about that a lot in later chapters. You've also got your cerebrospinal fluid, which is the fluid that surrounds your brain and spinal cord, so your central nervous system. And it helps protect the central nervous system, but also, again, plays an important role in terms of nourishment and getting rid of waste products from the central nervous system. And then you've got cellular secretions. So those include things like saliva and mucus. So a lot of your cells, for example, lining your gastrointestinal tract, so your stomach, your intestines, your esophagus, and your respiratory tract are going to secrete mucus to help protect themselves, but also to capture invading pathogens like viruses, bacteria, dust in the case of the respiratory tract, those kinds of things. Some of those fluids are going to act as lubricants. So while saliva does play a role in the digestive process, so for example, you've got some enzymes in saliva that break down carbohydrates. In addition to that, saliva plays a really important role in lubrication, helping food move from your mouth down your esophagus and into your stomach. We talked, if you've already had 201, we talked about the serous membranes pretty early in that class. So where that came up, the outer lining of your lungs and the inner lining of your rib cage both have a layer of cells called serous cells. And so what those do is secrete serous fluid. The membranes are called serosa, is the terminology I used in 201. And so those cells then secrete that serous fluid, which helps lubricate so that the lungs can slide nice and smoothly on the inside of the rib cage. So serous fluid, that lubricating fluid, would also fall under cellular secretions. Other things in terms of classes of extracellular materials, we've got the extracellular matrix, which as you can see there is the glue that holds cells together. So there's a number of different ways that we can attach cells together temporarily or permanently if we want to. And so one of the ways that we do that is with the extracellular matrix, and we'll talk more about that in a second when we talk about the glycocalyx. All right, but continuing with the concept of fluid compartments. So the two major fluid compartments are intracellular fluid, so all the fluid inside of the cells, and then extracellular fluid, all the fluid outside of the cells. And of course, fluid can change places. So water can move from one side of a membrane outside of the cell to inside of the membrane. If you're into exercise, which if you're in this class, you probably are, where you might have seen that, if you've ever gone to the weight room and lifted weights, particularly if you did a bunch of high repetition type training. So let's say you did a whole bunch of sets of, curls, or a whole bunch of sets of push-ups or something like that. And you notice that after doing, let's stick with curls, after you do that exercise that your elbow flexors, particularly your biceps, are a little bit bigger. So that's called transient hypertrophy, temporary muscle growth. And so what's happened there is the fluid from your blood plasma and some of that interstitial fluid from the force of the contraction, you've actually pushed some of that fluid inside of the muscle cells, and so the muscle cells are temporarily swollen. So you've moved some fluid from the extracellular compartment to the intracellular compartment of those contracting muscle cells. And so the fluid has changed places. It's moved from extracellular fluid in that case to being part of the intracellular fluid. So because this slide is titled the plasma membrane, I should probably talk about it. So the plasma membrane divides those two. So the plasma membrane separates the intracellular fluid from the extracellular fluid. So it is, as you can see there, an active barrier. So it allows certain things in, but it keeps other things out. So for example, water moves pretty freely, as we're going to talk about, but other things like sodium are going to be barred entry, except under certain circumstances. You also get things like glucose will be barred entry unless we help it move from one side of the membrane to the other. So it's a lot of the term I'm going to use repeatedly is going to be selectively permeable. It's going to allow some things in but keep other things out, or only certain amounts of some things like glucose in and then keep the rest of it out. So that's where it being an active barrier comes in. All right, so let's talk about membrane lipids. So here what we're talking about is the structure of the plasma membrane. So what makes it up? So in terms of lipids are fats. And so you're going to see a bunch of these things where it has lipid and something, right? So phospholipid is a phosphate head with a fat tail. And so your phospholipids are going to be the primary constituent of the cell membrane. And so what you've got here, so this is the close-up, so this is the cell membrane here. And then you've got your extracellular fluid out here. And then your intracellular fluid, the cytoplasm, in here. and then the cell membrane dividing the two. And so the cell membrane is formed from a phospholipid bilayer, right? So bilayer means two. So there's two layers of phospholipids, and each of these phospholipids has two components. So it's got a head, and those are these little dots here. And the heads of our phospholipids are charged. So they're polar is another term for that. So they have a little bit of an electrical charge to them. And so they are what is referred to as hydrophilic, which means that they are water-loving. So because the heads of these phospholipids like water, they face out into the extracellular fluid. And then on the inner layer here, these phospholipid heads face in toward the intracellular fluid. So the hydrophilic, water-loving heads face the water. The lipid, the fat tails, are hydrophobic. If you have a phobia, you're afraid of something. And so hydrophobic means you're afraid of water. Or in this case, our lipids are afraid of water. Our fats are afraid of water. And so they want to try to get away from it. And so our tails to our phospholipids face away from the water. And so if you've got two sets of tails, then they're going to, again, face away from the water here. So there's one set of tails, there's the other set of tails. And so because of these properties where the phosphate heads are polar, they're charged, and the tails are not charged and are thus hydrophobic, these particular molecules, these phospholipids, tend to sort of self-arrange into these bilayer type structures. So that those phospholipids, again, are going to make up the majority of our cell membrane. In addition to those, we're also going to have some glycolipids. So glycolipid is a combination of a sugar and a fat. We're going to have a few of those, and we'll talk about those here in a second. And then you've also got cholesterol. So, bring that in here. So these little yellow things are our cholesterol. And so cholesterol, its job is to help increase membrane stability. And so what they're going to do is they're going to kind of attach between these phospholipid tails and basically keep them from floating too far apart. Because remember that all of these molecules are in a fluid environment, and so they're going to tend to kind of float back and forth a little bit. And so when that happens, when these phosphate heads move apart, that would allow certain things to move inside of the cell. And so to minimize how much that happens, this cholesterol here helps hold those tails together to minimize the floating back and forth and keep those phosphate heads together to help stabilize the membrane and keep things from moving from one side of the membrane to the other without us intending for that to happen. So that's the job of cholesterol, is to help stabilize that membrane. So I left this video in here. Obviously, it's pretty old. It's from when I was in college. But I left this video in the slideshow, so if you want to watch that, you can do that. All right, so those are our membrane lipids, or membrane fats. Now we're going to talk about membrane proteins. And broadly, there are two categories of membrane proteins. There are integral proteins and then peripheral proteins. So if something is integral, it's really important. We can't get by without it, right? If something is peripheral, so think about your peripheral vision, that's off to the side. And so you could kind of get by without that, right? I mean, it wouldn't be ideal, but you could. And so integral proteins are proteins that are a structural part of the membrane that removal of them damages the membrane and can keep it from functioning. So integral proteins, we have to have them. Peripheral proteins are not permanently embedded in the membrane. Like sometimes they'll attach to the membrane and then detach. And so they are not, they're not structural parts of the membrane. So if we remove them from the membrane, it's not going to permanently damage it and change the way that the membrane functions. So integral proteins, have to have them. If there's damage to them, damages the membrane. Peripheral proteins, don't have to have them. I mean, they do perform important jobs in the cell. So for example, a peripheral protein, in your red blood cells, your cells have a cell skeleton, a cytoskeleton, which is largely formed of a protein called actin. And so cells can rearrange in shape a little bit. Like you can set up struts of actin to maintain one shape, or you can take them down and remodel them a little bit so the cell can compress some. And so those that actin, those are peripheral proteins that determine the shape of the cell. So you can move actin from one place to another, and it doesn't actually damage the cell's membrane. The cell's still able to function just fine. So in terms of integral proteins then, several different types. So you've got channels, which, trying to see where my channel is here. So this is a channel here. So a channel is kind of an open door. And so that's going to allow molecules, if they're the correct size, to move from one side of the membrane to the other. And then carriers typically physically change shape to allow a molecule to move from one side to the other. And so you can see here, this little carrier is going to change shape to kick out whatever this particular molecule is supposed to be. So those are our carriers. They physically move something from one side of the membrane to the other. And then enzymes are going to, when they receive some sort of signal from outside of the cell, they're going to start a signaling cascade inside of the cell that can initiate protein production or do a variety of other things. So that's the job of enzymes. I had some other notes here. I wanted to make sure I hit on those. So my additional note that I added earlier while I was looking at this stuff is that COVID binds to a transmembrane protein, which is an enzyme in this case, called ACE2 in order to gain entry into the cell. And you probably heard that in at least some of the early news stories. They talked a lot about ACE2 receptors, but specifically it's an enzyme. So if I say transmembrane, what that means is, so I'm going to use it in the context of a transmembrane protein. That means that is a protein that spans from outside of the cell to inside of the cell, or vice versa if you want to conceptualize it the other way. So it spans the length or the width of the cell membrane. That's a transmembrane protein. All right, the glycocalyx. So glycocalyx literally means sugar covering, and it is effectively the way that cells identify themselves. So the picture I've got for you in the lower right, these aren't human cells. Both of these are bacteria, but it's easiest to see on this one on the right. So this darkest line here is the cell membrane that we've just been talking a bunch about. So your phospholipid bilayer is there. And then this kind of fuzzy stuff here, a slime layer, that is this particular bacteria's glycocalyx. And so glycocalyx is a combination of glycoproteins. So glycoproteins are a combination of a sugar and a protein, and glycolipids, so a sugar and a fat, that project from the cell's membrane. And so they help the cell to identify itself, because different cell types are going to have different patterns of glycoproteins and glycolipids. And so a way to think about that is that the glycocalyx is kind of like the cell's fingerprint. So that serves a couple different functions. It helps our immune system determine self versus non-self. So this is one of our cells because I recognize this combination of glycoproteins and glycolipids, as opposed to something, if you get an organ transplant, that's a non-self organ. And so your immune system may attack that transplanted organ because it doesn't recognize it. It doesn't recognize the glycocalyx on some of those cells, and so it'll begin attacking that because it thinks that new implanted organ is an invader, right? So the glycocalyx serves as a way for the cells to identify themselves. So I mentioned both of these are bacteria, and you can see the one there on the left is a strep bacteria. But where we're going to talk about the glycocalyx when it comes up again will be in the digestive tract. So the cells, the epithelial cells, which again, an epithelial cell is a type of lining cell, and we'll get to that more with chapter 4. But we'll talk about the glycocalyx as it pertains to the gastrointestinal tract, but also as it pertains to the lining of the blood vessels, so in the cardiovascular system as well. Those lining cells have a glycocalyx, which helps identify, again, self versus non-self. It also helps with movement across the cell, so we see that particularly in the cells that line the blood vessels, which are called the endothelium. So if there's an injury or inflammation, then the glycocalyx can play a role in allowing adhesion of some of the immune cells in that area to help fight off the infection. Other things relevant to things like COVID that the glycocalyx does, so if we're talking about in the cells of the respiratory tract. So that sugar covering, that slime layer outside of the cell, plus the mucus that's actually produced by other cells helps trap viruses and hopefully keep them from making it inside of our cells where they'll hijack its machinery and reproduce and ultimately kill that cell. So also, while we're on the disease track here, so the glycocalyx presents one of the problems with cancer as well. So as you're probably aware, cancer is uncontrolled cell growth. And so what ends up happening there is you just make lots and lots of copies of this cell because there's a problem with basically the stop signal in the reproduction of that cell. So in a cancer cell, the glycocalyx can change almost constantly, which makes it really hard for our immune cells to say, okay, if you have a fingerprint that looks like this, attack that cell because that's a diseased cell. We need to make sure we attack that. Since the identification of the cell, since the glycocalyx is changing consistently, it makes it hard for our immune system to pinpoint, okay, we need to attack cells with this fingerprint. And so then that's one of the things that allows cancer cells to metastasize and move from their original site out into further areas of the body. All right, so one of the things I hadn't mentioned yet was that the glycocalyx can play a role in binding cells together. Well, I kind of mentioned it. So if we're talking about the endothelial cells, the cells that line the inside of your blood vessels, the immune cells can actually bond or attach to their glycocalyx if they need to. Again, for example, in the case of injury or inflammation. So one of the ways that cells can bind together is through the glycocalyx. Cells also bind together based on shape. So cells can be shaped in such a way that there's a convex surface on one where it curves out, and a concave surface on the adjacent one where it curves in, and so they can kind of fit together like that. Or they can have junctions. And so as you've got pictured here, this is kind of the intestine, the small intestine. So all the stuff you eat would be in here. So this would be the inside of the intestine, called the lumen, L-U-M-E-N, of the intestine. And then these are your little intestinal cells. And then all the stuff that you absorb from the lumen of your intestine, like sugars, will move through this cell, through the basement membrane, and then eventually into your bloodstream. So one of the things that our intestinal cells want to do, though, is to prevent things from moving between them, right? So we want to make sure that whatever gets in meaning into our bloodstream, is something that we want to allow in there. So, for example, if we had a disruption here, where these two cells pulled apart a little bit, well, things that were in the food that I'd eaten that may be bad for me, including things like bacteria, potentially maybe you didn't wash your hands before you ate, you got some viruses and bacteria in there that made it all the way into your small intestine. Then if you have a gap here between these two, that'll allow those to move between the cells and then into your bloodstream, and then they can get all over the body, and that would be a problem. So to keep that from happening, many cell types, including the cells of your small intestine, have what are called tight junctions. And you can see a close-up of a tight junction down here. So keyword here, those are impermeable junctions, so they don't allow things to pass between the cells, so they help us form a good solid barrier and keep things, again, from slipping between the cells. In the case of the small intestine, there's a condition that is still of some debate in the medical community as to whether or not it's a real thing, but the condition is called leaky gut. And so the idea with leaky gut is that something happens to disrupt these tight junctions and allow, again, viruses, bacteria, and a number of other organisms to migrate from the intestine into the bloodstream. And then those have been linked to a variety of nebulous systems ranging from things like headaches to acne or even more. So while that idea of leaky gut may or may not exist, one of the things that we do know is that there's a specific gut protein which was discovered in 2000 that was named zonulin, which is a fun name for a protein. And that protein, zonulin, is responsible for widening or tightening those gap junctions. So we do have the ability, based on the production of this protein, of zonulin, to open or close those junctions to make it easier or harder for things to pass between those intestinal cells. And it's likely that genetics play a role in production of that protein. Similarly, people who have celiac disease or Crohn's disease probably experience problems with their tight junctions based on the immune response. So you got some immune cells that effectively attack your own epithelial cells, your own cells lining the intestine. And so by doing that, that can weaken those junctions and allow things inside into the bloodstream, or can allow fluid to go the other way, and can play a role in producing some of the diarrhea and other symptoms that we associate bloating with those conditions. All right, and then desmosomes. You can see desmosomes here. So desmosomes, what they do is to help keep those cells anchored really closely to each other when there's a lot of shear forces. So shear forces would mean that this cell is being pulled up, this cell is being pulled down, and so that could, potentially pull them apart. And so the job of desmosomes is to basically Velcro those cells together and to keep them from pulling apart too much. So those oftentimes work in conjunction with the tight junctions. So we see desmosomes in cells where there's lots of mechanical stress. So we see that in the skin. There are desmosomes between cells in the skin to keep, so if something rubs against your skin or something pulls on your skin a little bit, you don't just get this massive separation of cells there. They stick together pretty well, owing in part to the desmosomes. We also see that in the myocardium, which is the muscle of the heart. So, your heart rate and blood pressure can both increase pretty significantly during intense exercise, sprinting or something of the like, weightlifting. So to keep those myocardial cells, to keep those heart cells from pulling apart, from pulling away from each other, they also have desmosomes, again, to keep them kind of Velcroed together. And then the last things are gap junctions, which are these channels between the cells that allow small molecules like ions, which are electrically charged particles, so things like calcium, potassium, sodium, potentially, depends on the type of the cell, but those can pass between those cells. So for example, gap junctions, sticking with the theme of the heart, play an important role in the electroconductivity of the heart, so normal electrical action in the heart to allow for orderly contraction. So we allow electrically charged particles to move between the cells of the heart so that we get orderly contraction of the heart muscle. All right, so back to the plasma membrane. So as mentioned earlier, the plasma membrane is selectively permeable, which means it allows some things in but keeps other things out. And not only that, it allows certain particular amounts of some things in while keeping the remainder of it out. And so there are two types of transport, two ways to get across the plasma membrane. The first is passive transport. And so the key thing to know about passive transport is that it does not require energy. And so examples there include diffusion and filtration. We'll talk about filtration when we get to the kidneys. We'll talk about diffusion today. Diffusion is when something moves from an area of high concentration where there's a lot of it to an area of low concentration where there's less of it. So diffusion happens passively. So again, there's no energy required. Something just moves from one side of the membrane to the other based on concentration, moving from areas of high concentration to low. Active transport, on the other hand, oftentimes we'll refer to it as pumping because it does require energy. So we have to use energy to pump sodium from the inside of the cell to the outside of the cell. We have to use energy to pump potassium from the outside of the cell to the inside of the cell. So both of those are examples of active transport. So when I say we use energy, I have the energy molecule pictured here in the lower right. So this is a molecule called adenosine triphosphate, or ATP. We're going to be talking about it a lot, especially later in the class. And so what we do is you have an adenosine backbone, and then you have three little phosphates. And the phosphates have high energy bonds between them. So there's one, and there's another one. And so we will break this bond between the last two phosphates with an enzyme, some sort of an ATPase. So for example, the particular ATPase in muscle cells is called myosin ATPase, and that enzyme is able to break this bond, and when we do that, that gives off free energy. And the energy that's given off then helps us perform work. So in the case of a muscle cell, that energy, when it's given off, helps drive muscle contraction. In the case of what we've been talking about here with the plasma membrane, if I need to pump sodium from the inside of the cell to the outside of the cell, that's going to require energy. And so I'll use ATP to drive those sodium and potassium pumps to push sodium outside of the cell. The reason it requires energy, should probably tell you that, is that you're going to be pushing sodium against its concentration gradient. So typically inside of a cell, especially let's think of a cell at rest, like a nerve cell at rest, inside of the cell you don't have very much sodium. Outside of the cell you've got a lot of it. And so if I want to push sodium from inside of the cell where there's already not very much to outside of the cell where there's already a lot, I'm pushing it against its concentration gradient. I'm pushing it from low concentration to high. And so doing that requires energy, as opposed to diffusion is where sodium goes the other way. So if I just opened up the door, if I opened up some channels in the plasma membrane that allowed sodium inside, sodium, given its preference, would move from an area of high concentration to low. It would move with its concentration gradient via diffusion. So it would just move from one side to the other, wouldn't require energy because it's moving from high concentration to low. So typically with active transport, where we're requiring energy, we're moving something against its concentration gradient. Speaking of diffusion. All right, so mentioned some of the concentration gradient a second ago. So what is diffusion? From a definition standpoint, diffusion is the tendency of molecules or ions, and again ions are electrically charged particles, to move from an area where they are in higher concentration to an area where they are in lower concentration. And the reason that happens, so think back to your high school, maybe even younger science classes that you took. Remember that molecules are in constant random motion. So they're all just moving around, right? And so if there's a whole bunch of them packed into a small space and they're all just moving around, they're going to bump into each other. And so given the opportunity, they want to spread out. And so that spreading out then is diffusion. And so that's what we've got down here pictured at the bottom. So you got this dye pellet in water. So you got a bunch of the little purple dye molecules all really close together. They're all bumping around. And so given the opportunity, when they move into a fluid environment, they're all going to spread out until hopefully, eventually, they're evenly distributed, right? So that is diffusion taking place, moving from high concentration to low until there's an even distribution, equal distribution throughout the solution. So how fast diffusion occurs is based on a few things. The first one is molecule size. The smaller the molecule, the faster diffusion occurs. So smaller molecules spread out faster. Also, the warmer the environment is, or the warmer the solution, which is the fluid, the warmer the solution is, the faster diffusion occurs as well. So in warm solutions, diffusion occurs quickly. In cold solutions, it occurs more slowly. So as it pertains to the cell's membrane, molecules can move from one side of the membrane to the other via diffusion, meaning they don't need help. They can just move through the membrane if they're fat soluble. So things like the fat soluble vitamins, which are A, D, E, and K, can move pretty easily from one side of the membrane to the other. But it also includes really small molecules like oxygen and carbon dioxide. Those can just move right through the cell membrane as well. Or they can move from one side of the membrane to the other if they're assisted. So if there's some sort of a channel, or let's go with that. If there's some sort of a channel, they're able to move from one side of the membrane to the other as well. So simple diffusion is kind of what I was mentioning with oxygen, for example. So oxygen just moves with its concentration gradient, and it actually goes on a pressure gradient, moves from an area of high pressure or high concentration to an area of low pressure or low concentration, all on its own. So that's simple diffusion, doesn't need any help. As opposed to facilitated diffusion, the molecule needs help to get from one side of the membrane to the other. So as an example there, you have transporters to move glucose, sugar, from outside of the cell to the inside of the cell. So there's different kinds of GLUT transporters. It's capital GLUT. So for example, one of them is insulin dependent. So after you eat a meal, your blood sugar goes up, pancreas releases insulin, and insulin in muscle cells, for example, has the effect of causing this signaling cascade that moves one particular type of glucose transporter called GLUT4 from the inside of the muscle cell to the cell membrane, so that that muscle cell can transport, it's a carrier, GLUT4 is a carrier, can transport glucose from outside of the cell to inside of the cell. So that then the muscle cell can use that glucose for energy, it can make ATP, or we can just store it as carbohydrate inside of the muscle cell as well. So facilitated diffusion means whatever the molecule is needs help to get from one side of the cell to the other. So from outside to inside or vice versa. All right, so facilitated diffusion. So we're going to talk primarily about channel-mediated diffusion. So, and specifically, we're going to spend most of our time talking about gated channels. So what you've got pictured here before I get to gated channels, so this is a leakage channel. And so you've got this transmembrane protein, so it runs from outside to inside. And a leakage channel is always open. So things are going to just move with their concentration gradient. There's a bunch of these little pink molecules out here. There's not very many of them here. So it's just going to diffuse through this doorway from an area of high concentration to low, from outside of the cell to inside of the cell. So that's a leakage channel. As opposed to a gated channel, which I apparently don't have a picture of, but that's okay. So we're going to talk primarily about two types of gated channels. There's a chemically gated channel, and then there are chemically gated channels, and then there are electrically gated channels. And so what those refer to is essentially the key to open the door. So a chemically gated channel is going to have a little flap here, a door, if you will, that's going to be closed until this protein receives a chemical signal. So for example, we see this when cells inside of the brain communicate. So one cell, one neuron, will release a neurotransmitter, say something like glutamate. And so it's going to release a neurotransmitter. When that neurotransmitter is received by chemically gated channels on the second cell, let's say there's a little receptor site there, then that's going to cause opening of the doorway, and that'll allow molecules to move inside. So that's a chemically gated channel. In response to receiving a chemical, like a neurotransmitter, that'll open up those channels and allow, often we're going to use this to describe things like sodium and potassium, will allow sodium to move inside of a cell, is a fairly common chemically gated channel. An electrically gated channel means that electricity is the key. So in an electrically gated channel, when the charge of the intracellular fluid changes, typically becoming more positive, then that's going to open up electrically gated channels. So that'll kick this door open and allow more sodium to come in, or we've got channels that go the other way and allow potassium to leak out. So electrically gated channels, change in charge of the cell membrane causes them to open. Chemically gated channels, receipt of a chemical, typically a neurotransmitter, causes them to open. All right, so last basic concept for today. So osmosis. So osmosis, probably the easiest way to think about it is movement of water from one side of the membrane to the other. But there's a few different terms on here. So there's solute and solvent. If you're anything like me, those were familiar when you had to know them for chemistry and then never again. So basically what those are, the solvent is the fluid, and the solute is the thing being dissolved in the fluid. So we're going to talk about, and my pictures show here, sugar water, right? And so these little red things are our glucose molecules. These are our little sugar molecules. And then obviously the rest of that is water. And so in that case, the sugar is the solute, it's the thing being dissolved, and then the water is the solvent, it's the thing doing the dissolving. So osmosis then, this is its definition, is the diffusion of a solvent, the fluid, through a selectively permeable membrane. So another important term on there is osmolarity. You can see the definition, it's concentration of a solute in a solution. And so if we've got a difference in osmolarity, difference in concentration of solute from one side of the membrane to the other, remember that, thinking back to that dye pellet, that our solute wants to disperse, wants to diffuse until the concentration of that particular solute is evenly distributed throughout the fluid, right? So we want equal concentration if we can get it. So for example, we're going to say this is inside of the cell over here. This is outside of the cell. Inside versus outside doesn't really matter here. This is our membrane, but we're going to say this membrane is freely permeable. And so what that means is that water could move from one side to the other, but so could our sugar. And so what's going to happen is the natural tendency is for that solution, that osmolarity, to balance out. And so because we've got a really concentrated solution over here inside of the cell and a relatively low concentration solution outside of the cell, if this membrane will allow sugar to pass from one side to the other, then our little sugar molecules are going to move from inside of the cell to outside until we get a balance of concentration from one side to the other. And now our osmolarity is equal inside of the cell versus outside of the cell. But of course, in our bodies, that's not usually how it works. So our membranes are selectively permeable, and they typically don't allow things like sodium or sugars, potassium to just pass from one side to the other unrestricted. And so what we have left then, because again, we want to balance out the concentration of solutions as much as we can, if we can't move the solute, then we'll move the solvent. And so what happens there is what's being depicted in this bottom picture. So we're at the same starting point down here in B that we were in A, where we've got lots of sugar here, relatively little sugar here. So we've got high concentration solution over here, low concentration solution over here. But now the new wrinkle is that our selectively permeable membrane won't allow the sugar to pass from one side to the other. And so what's going to happen then, if the sugar can't move, the water can. And so because we've got lots more sugar over here relative to the water volume, the water is going to move from this area of low concentration to the area of high concentration. In terms of the solute, I should probably clarify what I'm talking about. So there's a higher concentration of solute, of sugar, in this case on the right side than there is on the left, and so the water is going to move from left to right until we get a balancing out of the concentration of the solution, so until we get a balancing out of the osmolarity. A couple terms on there. So hydrostatic pressure is pressure that pushes water out, and so in this case, there is a hydrostatic pressure on the left pushing water out, and then pushing it into the more concentrated solution on the right. So hydrostatic pressure pushes water out, osmotic pressure pulls water in, and so on the right side here we've got an osmotic pressure pulling water in, trying to get it to balance out the osmolarity of the solution. So for the most part, water moves pretty freely across cell membranes. Part of that is probably due to, because again, water is a pretty small molecule. Remember I talked earlier about our phospholipids operating in a fluid environment and that they kind of float around, they move left, they move right. And so in doing that, that's going to allow small molecules like water to just slip from one side of the membrane to the other. But in addition to that movement, you also have aquaporins, which are channels that allow water to move from one side of the membrane to the other. So you get this combination of their channels that allow water movement, but then water molecules will actually slip between the phospholipid bilayer. All right, so why does any of that stuff matter? Because I know that gets pretty, the application of that's not super clear. So let's talk about why that would matter. So the case study I have for you is fairly old at this point. It comes from 2007. And in 2007, actually in January of 2007, we have a 28-year-old mother of three. Her name was Jennifer Strange, and she was found unresponsive on the floor of her Rancho Cordova, California home. So 28-year-old woman just found unresponsive and ultimately died a short while later. What could have happened? Well, what happened, or what's some important background information? The important background information is that hours before she was found unresponsive, she'd participated in a radio contest called Hold Your Wii for a Wii. And if you haven't seen a Wii, that's what they look like. I imagine all of you have, but anyway, that's what a Wii looks like. So it's a Nintendo gaming system. And so the contest was, who could drink the most water and go the longest without having to pee would win the Wii gaming system. And so the way this contest started out was that contestants drank 8 ounces of water, so a normal, like a juice glass of water, every 15 minutes for the first hour. And after the first hour, the volume that they drank increased, although I couldn't find any specifics on how much they drank after that. We do know that Jennifer Strange was able to make it an hour and a half or more into the contest, so she consumed well over 40 ounces of fluid, which isn't a ton, so it's more than a liter, but let's say she consumed somewhere in the neighborhood of potentially half a gallon of fluid at most before she bowed out of the contest. So she ultimately didn't win, unfortunately. But after the contest, she called in sick to work. And when she called in sick to work, she told her boss that she had a terrible headache. And so because of her terrible headache, she went home, and then that was the last time that anybody ever talked to her. But if somebody had been home when she got there, she probably would have reported other symptoms that include things like nausea, fatigue, eventually vomiting. So it kind of goes downhill from there. So you go from nausea and fatigue to eventually vomiting. You're going to have some muscular weakness, progressing to loss of consciousness and seizures, and then eventually death. So what happened? All she was doing was drinking water, right? So basically she drank so much water that it was ultimately fatal. And so what happened there is a condition called hyponatremia. And so by drinking too much plain water, i.e. water, not Gatorade that also has things like sodium, potassium in it, by drinking too much plain water, effectively what you end up doing is actually flushing out a bunch of the sodium from the extracellular fluid. And I have a really scientific, really professional MS Paint depiction of this. Obviously, normally in class I would do this on the whiteboard, but we don't have one, and I don't have a better idea, so I'm going to do this in Paint. So what you've got here is you've got obviously the inside of the cell here, and so each side has the same concentration of solute. We're going to call this sodium. So the little blue dots are sodium. So there are, I think I made 13 on each side. So there's 13 little sodium molecules outside of the cell, 13 little sodium molecules inside of the cell. Well, if you go and drink a bunch of plain water, then we start getting rid of sodium molecules, right? You flush them out. You actually excrete them. So you lose more and more sodium until you end up with a really concentrated solution inside of the cell and a really low concentration outside of the cell. Well, think about what we just talked about in terms of water movement. So you've got low concentration out here, high concentration in here. Sodium can't just move from one side of the membrane to the other, so these little molecules can't just move from inside of the cell to outside because our cell membrane is impermeable to them. So what's going to happen is water is going to move one way or the other. And so remember that the water is going to try to move from outside of the cell, in this case, to inside of the cell to make the concentration the same. And so you're going to get this massive movement of water from movement of water from outside of the cell to the inside of the cell to try to balance out the concentration of sodium molecules. And so when that happens, when a bunch of water moves inside of the cell is going to swell. When that happens in the central nervous system, so in the brain, you're going to get a swelling of your neurons, of your brain cells, and then you get a swelling of the brain on the whole. Well, obviously your brain is encased in your skull, so when the brain starts to swell, it can't really push out, and so what it does instead is to, tries to push out against the skull, but ultimately then it starts to push down on itself, and particularly it pushes down on the lower part of the brain stem, which is kind of right there at the top of your spinal cord. When that happens, you've got your respiratory center, where you initiate the signals causing you to breathe, and then your or the ventilatory center. And then you've got your cardiovascular center that regulates heart rate there as well. Both of those are in the brainstem. And so when the brain swells and pushes down on the brainstem, then that compresses those centers, ultimately results in stopping breathing and stopping the heart from beating. And so then that becomes fatal. So effectively what happened was she drank so much water that she got rid of all this fluid outside of the cell, made her extracellular fluid really dilute, or really low osmolarity. And so then that caused water to move inside of her cells to try to balance those concentrations out, cause cell swelling, and then brain swelling ultimately resulted in death. And so what I'm talking about there is a concept called tonicity. And so tonicity, you can see the definition, is the ability of a solution to change a cell's shape. So if we have an isotonic solution, you can see that means that it has the same concentration of non-penetrating solutes. So the same concentration of sugar, sodium, etc. as we would find in intracellular fluids. So if you put immerse a cell in an isotonic solution, you're not going to get any movement of water either way, because both inside of the cell and outside of the cell have the same concentration of non-penetrating solutes, so again, sugar, sodium, potassium, etc. If we place a cell in a hypertonic solution, so if we take a cell and put it in put it in a solution that has lots and lots of non-penetrating solutes, then what's going to happen is, so if all the fluid out here has lots of sodium, for example, then the intracellular fluid is going to try to balance that out, and it's going to leave the cell. And so then the cell is going to shrink. So where that comes into play is, for example, you're all probably at least peripherally aware that you can't drink seawater. So if you ever get stranded on a raft in the middle of the ocean, you can't just drink ocean water. And the reason for that is because ocean water is hypertonic. It has lots and lots of sodium in it. That's why it has such a salty taste. And so if you drink a bunch of ocean water, that really concentrated salt water inside of your intestine is going to cause fluid to move from your intestines and from the extracellular fluid deep to that. That fluid is going to move from the inside of your intestines, it's going to move into the lumen, the very inside of the intestine, to try to balance out the concentration of that salt water. So effectively, by drinking the salt water, you're actually going to give up fluid to try to dilute the salt water. And so drinking ocean water then is actually dehydrating. It pulls fluid out of you and puts it into the lumen of your intestine. And then that can cause diarrhea, etc. So that's why I can't drink salt water or can't drink ocean water. And then a hypotonic solution is the opposite of that. So it is a solution that doesn't have any or doesn't have as many non-penetrating solutes. So for example, what we just talked about with the hyponatremia, what ended up happening is that excessive water consumption makes your extracellular fluid hypotonic. So you flush out those non-penetrating solutes, and so then that pushes fluid into the cell and then causes cell swelling, which is what you see here. So all these are red blood cells, but you get the basic concept if that were happening in the central nervous system. All right, so that's it for lecture 1. So I'll work on lecture 2 and try to get that posted as soon as possible. And so we will see you next time.